Hi guys, welcome to my video DNA structure on a molecular level. So this video is kind of go over the half of week 2 where we actually zoom in on what does a DNA molecule actually look like on an atomic and molecular level. In week 1 we talked about an entire chromosome and what an entire chromosome looks like, and this week we're going to kind of zoom in and see what does the DNA actually look like, because understanding this is going to really help you out next week and in the weeks after that when we talk about how DNA is replicated. Understanding the structure of DNA is actually going to help you a bunch, because the structure really does determine how we work with it. So understanding this on a good level is important for future weeks. So we'll go over it in detail here so that I can hopefully help you understand it well enough to feel comfortable with it, so that it doesn't sabotage you during the rest of the semester if you don't understand it well. Okay, let me erase this here. Okay, here we go. DNA. So we talked about this during week 1 here, but DNA stands for deoxyribose nucleic acid. Or sometimes they will say deoxyribo and leave out the SE. Occasionally you will see deoxyribonucleic acid. That can be a way you see it too. Sorry, I should lowercase this r because it's not in, there isn't an R in DNA. Deoxyribonucleic acid or deoxyribose nucleic acid, you'll see it both ways. They mean the same exact thing. When a lot of people think of DNA, they think of the traditional little twisted ladder picture here, where you get something like this. Oh, that's beautiful right there. It's a good thing I'm not an art major, right? This is kind of what people picture when they think of DNA, and that's true. This is the idea of what DNA looks like. But we're going to kind of zoom in and get further in on this. And we'll show you where that shape comes from. DNA, just like everything else, DNA is made up of building blocks, of pieces that we assemble together to make a DNA segment. These little pieces are called nucleotides. These are DNA building blocks. A nucleotide is made up of 3 pieces. There is what we call a sugar. Its more formal name is a pentose sugar, and I'll show you what that means later. Really what it is it's just there's 5 carbons in it, so it actually is shaped like a pentagon. And I'll show you a picture of that later. You don't need to know that, but that's where the name pentose comes from. It's a pentose sugar, or sometimes we just call it the sugar. So a pentose sugar, and poking off of it on one side here is what we call the phosphate. If you've ever heard of phosphorus, which is like an element, sometimes they use it in bright flares and things like that. There is a phosphorus atom in that molecule there, and so that's where the phos part of this comes from. Is it's phosphate, named after the phosphorus that's in it. So so there's the phosphate poking off of one side of the sugar, and then poking off the other side of the sugar is what we call the nitrogenous base. Sometimes it's just referred to as a base, but the official name is the nitrogenous base. We call it the nitrogenous base because it has lots of nitrogen in it, and that's the only place in an entire DNA nucleotide that you see nitrogen. So that's where the name comes from. You don't need to know that, but that's where the name comes from. Is we call it the nitrogenous base for that reason. but oftentimes we'll just refer to them as bases as well. So be prepared to see it either way. So these three components stuck together like this whole thing is a DNA nucleotide. Now, when we want to hook two nucleotides together, remember DNA is a big long string. So we're going to build it just like a chain. We're going to hook a bunch of these things together to make a big long string. When I want to hook the next one in here, so the next one's got a phosphate and the sugar and a nitrogenous base here. Connect those on. Okay, so this would be the next base in the chain. The way that we connect that on is we take this phosphate on this nucleotide and we're going to hook it to the bottom of the sugar on the previous nucleotide. So notice we've got this sugar connected to its phosphate, and then its phosphate is connected to the next sugar. And then if we were to add yet another nucleotide on here, or another one, nitrogenous base is poking off here. Then we would have this phosphate connected to that sugar, and this phosphate connected to that sugar. So notice, and we would just keep doing this over and over and over again, build a really big long chain of these nucleotides connected together by their phosphates and sugars. So notice what this does here is it gives us a strand where we've got phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, over and over and over again, all connected together. And the nitrogenous bases are poking off to the side here. You see that? So notice the nitrogenous bases are only connected to their sugar. These lines here, these are called covalent bonds. Let me write that word out here. These are really strong bonds. They're glue, they're permanent. This is like getting sealed in the temple kind of thing. So the nitrogenous base is permanently glued to its sugar. And the sugar is permanently glued to its phosphate kind of thing. Like that's what those lines are, is they're really strong permanent bonds. They're covalent bonds. And so when we glue this phosphate to that sugar, it's really, really hard to get it back apart again. But we can do it, it's just really, really hard. And we'll talk about that later, in later weeks, I mean. So these are really, really strong. Nitrogenous base is glued to its sugar, and each sugar is glued to its phosphate, and we've got these big, strong covalent bonds down the back here. Notice this part right here, these sugars and phosphates kind of form like a rod. And this is what we call the backbone, the DNA backbone. You might be able to see where we get that name. It's kind of like your spine with the ribs poking off the sides sort of thing. Like, so we've got, so the sugars and the phosphates together make up the DNA backbone, and the nitrogenous bases poke off into the middle. And then a phosphate, a sugar, and a nitrogenous base make up one nucleotide. So this right here is one nucleotide. And then this column right here of the sugars and the phosphates all connected together, that was the DNA backbone. Okay? So good so far? I mean, I guess you can't answer, huh? Tutoring habits. Oh yeah, here we go. Let's just back that up here. This will be easier. Okay. I'm gonna erase this back here to give me some room to work. Well, we learned this week that DNA is what we call a double-stranded molecule. What that means is it's actually two strings that are next to each other. And so the way that this works is we're going to have another strand over here, another string of these sugar phosphate nucleotides with the base kind of poking off here in the middle, where we've got a sugar right here. I'll just draw out a bunch of these sugars here. A bunch of these sugars. Okay. phosphates. There, connected to 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 there. Okay, we've got our nitrogenous bases poking off in the middle here. Okay, so this is kind of how this works, is these things are parallel to each other. So we wind up with these two strands of DNA that are parallel to each other, and this is what forms double-stranded DNA. And if you use your imagination, you can see where we've got a sugar phosphate backbone down here. If you were to simplify it, you can make it a line. Sugar phosphate backbone over here, you can make another line. And then you got your nitrogenous bases going across the middle. And it kind of looks like a ladder. And if you were to twist this, and I'll show you later that that's how DNA is, you twist it, you kind of wind up with a twisted ladder shape, which in 2 dimensions kind of looks like that. That's where you get your DNA shape, is this is what's really going on, and then we'll twist it. And again, I'll show you a 3D image of what it really looks like. But that's where the twisted ladder shape comes from. So when you guys are making your DNA models out of candy this week, make sure you get your phosphates and your sugars and your bases, and make sure that you remember that your backbones are made up of alternating phosphates and sugars. Your bases are poking off in the middle, but make sure you remember that bases don't poke off of phosphates. That's probably the biggest mistake I see people make on their DNA molecules, is when they're building their DNA molecules, yeah, they alternate sugar and phosphate down the side, but then they accidentally put little nitrogenous bases off of the phosphates as well as the sugars. And that's incorrect, and you will get docked points for that. So make sure that you only have nitrogenous bases poking off of the sugars, not the phosphates as well, because the phosphates are only connected to sugars, not nitrogenous bases. Okay, across the middle here, because somehow we have to hold these 2 strands together, we've got we're not going to covalently bond these together, because covalent bonds are really strong, and we'll learn next week that I want to be able to separate these strands, right? So I don't want the sides of the ladder to break, because that'll just mess up the whole sequence, which is a really bad thing. So we want those to be held together with those strong covalent bonds, but across the middle, I just want them to be weak so that I can pull them apart and put them back together again at will, because we'll find you need to do that a lot in weeks 3 and 5. We separate the strands and put them back together again all the time. So we do want to hold these together, but they can't be covalently bonded together, because then it would be really hard to pull them apart. So instead, we're going to use a weaker bond called a hydrogen bond. A hydrogen bond. So these are weak bonds. These are kind of like magnets. So a covalent bond was kind of like glue or welding. Hydrogen bonds are like magnets. You can stick 2 magnets together and they'll hold, but you can easily pull them apart. Depending on how strong the magnet is, it determines how easy it is, right? But you can easily pull the magnet apart. So if we kind of were to magnetize these 2 strands to each other, then they would stay together when I wanted them to, and I could pull them apart when I wanted to. It makes it more functional. So these nitrogenous bases that are poking across the middle are going to be hydrogen bonded to each other. They're like magnetized to each other. So we'll have these little attractions across the middle here. Again, it's not like solid hard bonds. These are not covalent bonds. These are just like magnetic attractions across the middle here. Those are called hydrogen bonds. And we'll find that they're going to make different numbers of hydrogen bonds, and we'll come back to that. But that's how they're held across the middle. So you got hydrogen bonds going across the middle, where the nitrogenous bases on opposite strands are going to be kind of paired up with each other and held together with these hydrogen bonds. And then nitrogenous base is covalently bonded to its sugar, and the sugars are covalently bonded to their phosphates up and down the backbones. Okay, So we'll come back to the hydrogen bonds here in a sec. There's a detail here that I want to get into first that will make the hydrogen bond conversation better when I make the jump up. Let's focus in on these nitrogenous bases. It turns out that there are 4 different kinds of nitrogenous base that we find in DNA, and we give them specific names. The 4 different kinds of nitrogenous base are called adenine, cytosine, guanine, and thymine. Those are the names of the 4 nitrogenous bases. So these are nitrogenous bases. So each of them performs the job of a nitrogenous base, but each one's ever so slightly different, and there are little details for each one that we do expect you to know. So let me zoom in on these guys, and I'll show you what those details are, and then I'll teach you my way of remembering them, because it can be a little tricky to keep track of all of the little details as to what's special about each of the 4 DNA nucleotides and how you can tell them apart. The 4 DNA nitrogenous bases and how you can tell them apart. So let's go and look at that. So I'm going to use my little 3D simulator here. What this is going to show is what a DNA nucleotide looks like on a molecular level in real life with atoms and things like that. It's going to look really complex and scary. You won't need to know it in this much detail. And I'll orient you with what you need to know on this picture. The reason why I pull this up is it's going to help me show you the structure of the nitrogenous bases. And it'll help make that a lot more sense for you visual learners that want details. So let me pull up an adenine right here. So here's a nitrogenous base. And again, this looks really scary. Orientation here, the spheres are atoms. The sticks between them are covalent bonds, okay? The different colored spheres represent different types of atoms, and you don't need to know what those are here. Okay, as your orientation, this little orange and red firework right here, that's your phosphate, okay? This crazy little blue and black thing out here, that's your nitrogenous base. And if I pivot this here, pivot this right here, this little black and red pentagon right here, well, that kind of highlighted part of the phosphate over there too, but this little pentagon right here is the sugar, okay? So there's your 3 parts. You got your nitrogenous base, your sugar, your phosphate, okay? So it's not as bad as it looks. This is just what the atoms look like, and you don't need to memorize the atomic structure here. You don't need to know which atoms are where or whatnot. The whole goal here is simply to show you details here on the nitrogenous base. Okay, notice here on the nitrogenous base, we've got these 2 little circles right here. There's one right there, there's one right there. They're like rings. We got these 2 rings right here. This is adenine is what we call a purine. And a purine means that the nitrogenous base has 2 rings in it. And when we say 2 rings, that's what we mean is if you look at the physical structure atom for atom of the nitrogenous base, you can distinctly see there are 2 circles of atoms here. I mean, it's a pentagon and a hexagon, but imagine they're circles, right? So we've got a ring right here and a ring right there. There are 2 rings in the nitrogenous base, and when there are 2 rings, it's a purine. And I'll show you a little trick to remember that here in a sec. So adenine is an example of a purine because it has 2 rings in its nitrogenous base. Let me pull up a different one. So that was adenine. Let me pull up cytosine here. Okay, so again, we're looking at it. Here's the phosphate down here. Here's the sugar right here. It was a pentagon. I won't bother turning it on its side so that you can see it. But then here is the nitrogenous base. Now, if you look at this, the nitrogenous base is different. What's different about this one? Well, it doesn't have two circles. It only has one ring right here. So it's not a purine because it doesn't have two rings. It only has one. This is what we call a pyrimidine. I know that's a tongue twister. Pyrimidine equals one ring in the nitrogenous base. So if you've got two rings, you're a purine. And if you got one ring, you're a pyrimidine in your nitrogenous base. Of the four nitrogenous bases, two of them are purines and two of them are pyrimidines, and you will need to know which ones. So I will show you a little trick for remembering those. Let me back here. Okay. Remove that to give me a clear whiteboard here. Okay. Pop that over here. Clear my drawings. All right, here we go. Okay, here's my little trick here. If you list out the four bases, so often we abbreviate the bases by their first letter. So if you list out the first letter of the four bases alphabetically, so you go, so here's my method of remembering nucleotide features, or sorry, base features. Let's say base features. Base features. So here's my little way of remembering this, okay? If you list them out alphabetically, you've got A, C, G, T, right? Okay. A is the first letter of the alphabet, so it comes first, and A stands for angel. Again, it actually stands for adenine, but bear with me. It stands for angel, okay? A stands for angel, and angels are the holiest beings in all the universe, right? They are just super, super holy. And often we picture angels with halos. I highly doubt they have halos, but, you know, bear with me here. Angels have halos. Well, the halo usually means you're holy, right? So the more halos you have, the holier you are, right? So if you got one halo, man, you're doing good. But if you got two halos, you are like exalted here, right? Like I'm imagining like exalted people just having this pillar of halos on top of their head. It would look really funny. But angels are the holiest ones, so they get two halos, not just one. They get two halos. And angels are extremely pure individuals, and so they are purines. So adenine has two rings in its nitrogenous base, and it's a purine. That's how you can remember that one. Is angels are super holy, so they've got two halos, not one, and they're pure. Then it turns out if you alternate down the alphabet, they actually bounce back and alternate two rings, one ring, two rings, one ring. So we've got adenine has two rings and it's a purine. Cytosine has one ring, which makes it a pyrimidine. Guanine has two rings, which makes it a purine. And thymine has one ring, which makes it a pyrimidine. So if you can remember that A stands for angel, which means it's got two halos because it's super holy and it's a purine, and then you can remember your alphabet, just alternate down the bases. So C has one ring, G has two rings, T has one ring. And that can help you remember which ones have two rings and one rings and which ones are purines and pyrimidines. Purine means two rings, pyrimidine means one ring in its nitrogenous base. Okay, we'll come back to this here in a sec. These bases, remember, we've got to have them pair up across the middle, and they actually have preferences for who they pair up with. Adenine always pairs with thymine. So A and T go together all the time. And C and G go together all the time. You never have an A pairing with a G or a C pairing with a T or an A pairing with a C or a T pairing with a G. You never have that. A always pairs with T and C always pairs with G across the middle, okay? So notice this is just the hydrogen bonds across the middle. Anytime you've got an A nitrogenous base on one strand, there must be a T on the opposite strand facing it. That's what that means. And anytime you got a C on one strand, you got to have a G facing it on the other side. So A and T, C and G. Now, if I come and pull this up here, let me actually pull up a picture here. Just to show you, just to make a point here. Notice that, so here's pictures of the 4 DNA bases in my little molecular model thing. Notice the phosphates and the sugars are all the same on all 4 of them. The difference between them is their bases. And notice down here, so here's their nitrogenous bases from overhead. Notice A has 2 rings, C has 1 ring, G has 2 rings, T has 1 ring. So there you go. You got purine, pyrimidine, purine, pyrimidine. And then notice the other differences are these molecules poking off the side. What atoms do you have poking off the side? And that's what makes you a cytosine 1 ring as opposed to a thymine 1 ring. But you don't need to know the things poking off the side. Just know that adenine has 2 rings, cytosine has 1 ring, guanine has 2 rings, thymine has 1 ring. The other feature is how many hydrogen bonds each of these bases makes. Okay, so remember we talked about these hydrogen bonds that pair up across the middle, right? So adenine and thymine make 2 hydrogen bonds between each other. So if this was an A base right here, so let's say that was an A, and this one's a T, then they're going to make 2 of these little hydrogen bonds in between them, okay? And if you've got a C and a G, C and G make 3 hydrogen bonds between them, okay? So think of it as 2 magnets holding A and T together, 3 magnets holding C and G together. So A and T have 2 hydrogen bonds, C and G make 3 hydrogen bonds, okay? So that's the other differences is, so you can always tell a DNA base apart by how many rings does it have and how many hydrogen bonds does it make. Because A has 2 rings and makes 2 hydrogen bonds. C has 1 ring and makes 3 hydrogen bonds. G has 2 rings and makes 3 hydrogen bonds. And T has 1 ring and makes 2 hydrogen bonds. Let me show you a way to remember those. We'll add on to my little chart here. Make room here. Okay. A pairs with T. And if you've ever heard of the telephone company AT&T, right? So that helps you remember the A and T pair together is because you got AT&T. And there are 2 T's in AT&T. So A and T make 2. A and T make 2 hydrogen bonds. And C and G, if I were to spell out C and G, you got C and G make 3 hydrogen bonds. So that's a way to remember it is you got AT and T, A and T make 2, C, G and 3, or C and G make 3 hydrogen bonds between each other. So A and T make 2 hydrogen bonds, C and G make 3. So if you can remember that, or if you can remember this little diagram here, you'll always be able to remember how many rings each of the 4 bases has, whether it's a purine or a pyrimidine, and how many hydrogen bonds it makes, and who it pairs with. So you can nail all 4 of those features just by remembering this little mnemonic, and you're welcome to toy around with it to remember it in your own special way. This is just one way to remember it. It's not the only way. But that's a way that you can remember which ones are which. Now, why does A always have to pair with T and C always have to pair with G? There are 2 reasons for that. One we already covered. If I were to try and pair A with G, A makes 2 hydrogen bonds, G makes 3. Those don't work. Okay, so that's one reason why you can't have A pair with G or A pair with C, is they don't make the same number of hydrogen bonds. The other reason why that won't work is in their rings. Notice A, so A always pairs with T. Notice how many rings does A have? A has 2 rings. How many does T have? T has 1. If I've got C paired with G, C has 1 ring, G has 2. Notice an A and T pair is 3 rings across. A C and G pair is 3 rings across. So notice by having A pair with T and C pair with G, the DNA molecule stays a uniform width all the way down, because all of the pairs are the same width across. They're all three rings wide, because you always have a 2 ring paired with a 1 ring, a purine paired with a pyrimidine. You always have a purine paired with a pyrimidine. And then it's the number of hydrogen bonds. So like A could hypothetically still pair with C and give us 3 rings across, because you'd have 2 rings paired with a 1 ring. But C makes 3 hydrogen bonds, A makes 2 hydrogen bonds. So those 2 factors are what determines what makes A pair with T and C pair with G, is the number of rings, because you always need to be 3 rings across in total, and the number of hydrogen bonds. If you had T pair with C, you'd have a little divot. And if you had A pair with G, you'd have a bump. And you don't want ladders that are divoting and bumping. You want them to be uniform width all the way up. So there you go on that one. That's the story on the nitrogenous bases. Now let's go look at the sugars. It turns out that these DNA strands are what we call antiparallel. Let me write that word out here. Antiparallel. No, I think it's 2 L's in the middle. Parallel. There we go. I always spell that word wrong. Antiparallel. What this means is you're parallel, which means you got 2 lines that are you've got 2 lines that are next to each other running in the same, they're side by side, they don't intersect, right? This is math. Parallel. Anti means opposite, which means they are pointing in opposite directions, kind of like a highway. You know how if you've got a highway, traffic on one side of the road travels one direction, and traffic on the other side of the road travels in the other direction. The 2 lanes are parallel to each other, but they are traveling in opposite directions, hence they are anti-parallel. DNA works the same way, and I'll show you what I mean by that. If we pop back over here, let me show you a double-stranded version here. Okay, so here's an A paired with a T, right? So we got our adenine one right here, and here's our thymine one right here, and our hydrogen bonds are invisible here across the middle, okay? So there's your thymine nucleotide, your adenine nucleotide down here. All right, and again, you don't need to be able to recognize these by sight, but if I did show you these guys have 2 hydrogen bonds right here, then you would know, well, this one's got 2 rings and makes 2 hydrogen bonds, so that must be adenine. This one's got 1 ring and makes 2 hydrogen bonds, so that must mean it's thymine, right? So you wouldn't even need to recognize the structure. You just need to know how many rings there are and how many hydrogen bonds it has, and you know exactly which base it is. Okay, anyway. If I flip this on its side, so we can look at the sugars. Remember, the sugars were these little pentagons here. Notice this sugar, this pentagon here, oh, pentagon right there. If you use kind of the little point here as an arrow, you'll notice that this pentagon's kind of pointing that way. You see that? Like if you pretend it's an arrow, it's pointing that way. This pentagon over here on the opposite base, right here, notice it's pointing the other way. The pentagons are upside down from each other, or backwards. So the pentagons on this strand are pointing that way, and the pentagons on that strand are pointing that way. So the strands are parallel to each other. They're running alongside each other, but they're actually pointed in opposite directions. And that's what the sugars can help you see. Now, in order to help us keep track of which strand is pointing in what direction, we have these labels that we're going to put on here. And let me bounce back over here. So if I've got two strands of DNA, one strand here and one strand here, and they're running next to each other, we got our nitrogenous bases across the middle. Okay. We've got to name the ends of these things. Okay. So you've got to have a front end and a back end for each strand. The names for the front end and the back end, we call them 5', which means 5 prime. We say that 5 prime. So you would pronounce it like 5 prime. That's what 5' means, is 5 prime. So one end of the strand is named 5 prime, and the other end of the strand is named 3 prime. Okay, so these are names that scientists came up with, and I'll show you here in a sec where the names 5 prime and 3 prime come from, and how you can tell the 5 prime end apart from the 3 prime end. But because these strands are antiparallel, what that means is if the 5 prime end of this strand is on the left side, that must mean the 3 prime end of the bottom strand, the opposite end of the bottom strand, is going to be across from it. So the 5 prime end of the top strand is on the left, the 3 prime end of the top strand is on the right, the 5 prime end of the bottom strand is going to be on the right, and the 3 prime end is going to be on the left, because they have to be traveling in opposite directions. So that's what we mean by antiparallel, is the strands are parallel to each other, but 5 prime and 3 prime, the ends of them are pointed in opposite directions. They're traveling in opposite directions, just like a highway. That's what antiparallel means. Now, how can you tell the 5 prime end of a strand apart from the 3 prime end of a strand? Let me show you. This goes back to our DNA nucleotide. So let me pop back over here. Let's go back and look at our sugar here. So let me flip this on its side. Spin it around here to where it's at the same orientation we've been working with. Okay, here we go. So here's our sugar. So we got our little sugar pentagon here. Okay, so here's the phosphate right here. Okay, that was our phosphate. I can spell phosphate. Over here, this is our nitrogenous base. And right here, I'll make it white to stand out from the black background here. Right here, this is our pentose sugar, okay? So let me zoom in here on that pentose sugar. We're going to focus here. When scientists are trying to, when scientists are trying to write about these molecules in their scientific papers for their fellow colleagues and talking about inventions and things like that, they often have to refer to specific atoms within the molecule. And it's really, really awkward in a scientific paper to try and be like, well, you know, I'm talking about the third carbon from the third carbon on the sugar when you start at the top and you go clockwise around, it's like the third carbon down kind of thing. We worked with that carbon atom. It's like there was a whole paragraph there where you're like, I'm so lost. So rather than having to describe where our atoms are, we gave them names. So I can be like that carbon atom and everybody knows what we're talking about kind of thing. So we gave the carbon atoms. So carbon atoms in my picture are the dark gray black one kind of thing. Okay, so anytime you have a dark gray or a black sphere here, that's what I meant, the black sphere, that's a carbon atom, okay? So notice there are 5 little carbon atoms here in the sugar. There's 1, 2, 3, 4, and then there's a 5th one. It's kind of hiding behind the phosphate, but it's back there, okay? So there are 5 carbon atoms, hence why it was a pentose sugar. 1, 2, 3, 4, and then a 5th one kind of out on a stick over here, okay? And I might even be able to get rid of the phosphate here so that we can see better. So I'm just getting rid of the phosphate so that we can see back there better. Okay, so remember there is still a phosphate, but I'm just zooming in here on the sugar. Okay, so there you go. So there's your 5th carbon out there. Okay, so we gave them names. The carbon that is connected to the nitrogenous base, because here's our nitrogenous base out here. So the carbon that was connected to the nitrogenous base is called 1'. We just decided to name him 1'. Okay, and then we went clockwise around the circle here. So we've got 2'. That's the 2' carbon. This one right here is the 3' carbon. This one right here is the 4' carbon. And this one right here is the 5' carbon out on the flag here. Okay, so those are their names. We named them 1', 2', 3', 4', 5'. And then of course, remember your phosphate is poking off of this one here. Okay, your phosphate is hanging out over here. Okay. Now that we've named those, you can see, so again, you don't need to know this. I'm just explaining where 3' and 5' come from because a lot of students wonder about that. Notice this 3' one, this 3' carbon has a little oxygen, oxygen's red, poking off the bottom here. And the 5' one has the phosphate poking off of it. Notice that? 3' pokes off there, 5' pokes off there. Well, on our little model of a DNA strand that we made over here, notice that we've got an end of the strand where we've got a free phosphate that's poking off the end that isn't stuck to the next sugar in line. You notice that? We've got a phosphate that's just hanging out by himself here. And down here on the other end, we've got just a sugar that's not connected to the previous phosphate kind of thing. You notice that? So because the strands aren't, or the nucleotides aren't symmetrical, we're going to have one end of the strand where there's a phosphate poking off and one end of the strand where there's just a sugar poking off. Because the phosphate's connected to our 5' carbon, the phosphate, or the end of the strand that has the phosphate poking off of it is called the 5' end of the strand. Because it's the end where the phosphate is hanging out to dry. And the other end of the strand is the one where the 3' carbon's little oxygen group is hanging off the bottom instead. And so that is called the 3' end. So in summary, the end of the strand where the phosphate is exposed, that's the 5' end. The end where the sugar is exposed, that's the 3' end. And notice on the opposite strand here, the end with the sugar exposed is over here, and the end with the phosphate exposed is over here. So again, they're anti-parallel. The 5' end of this strand is opposite of the 3' end of that strand, and vice versa. Okay? So if I gave you a strand like this, because you know that 5 and 3 the strand is anti-parallel, and the 5' end of a strand is always opposite of the or is always on the other end of the 3' end. If I were to ask you a question on a quiz or an exam or something where you've got like the 5', I said, there's the 5' end right here. What are the other 3? You know that this has to be the 3' end of that top strand because that was the 5' end. And you know that the bottom strand has to be backwards. So you know that one's going to have to be the 3' end and that one has to be the 5' end. So if I gave you one of the 4, you would be able to fill out the other 3. Okay, and note, 5' and 3' are, we call them 5' and 3', not left and right. Left and right are completely arbitrary terms. So people get into the bad habit of thinking 5' is always going to be in the upper left-hand corner. That is not true. This, if I were to draw it like this, that is a perfectly valid DNA molecule. They're anti-parallel. 3' is opposite of 5'. So remember, left and right are completely arbitrary terms. Okay? So the 3' end of a strand is always opposite of the 5' end, but the 5' isn't necessarily going to be in the upper left-hand corner. So just remember that. Okay. So there's 5' and 3'. Trying to think what else I am missing here. Okay, if we go and look over here, there was one question that one point that I made at the beginning that I never came back to. So here's my AT pair again. Notice that we've got, let me actually add in the hydrogen bonds here. Okay, so here's these little green lines represent the hydrogen bonds across the middle. Okay. If I add the next pair, so let me make it a CG pair. Notice what I did there, if I turn this on its side, there's our phosphate, sugar right there, right? The phosphate and the sugar of one nucleotide, the phosphate and the sugar of the next nucleotide. We've built our DNA backbone there, our DNA backbone there, and then the DNA bases are going across the middle. The reason why we always show it as that little twisted ladder shape is if you'll notice here from the top, the DNA nucleotides don't stack straight on top of each other. They're kind of rotated ever so slightly. If I add the next pair right there, notice it's not directly on top, it's slightly turned. And if I add the next pair, it's slightly turned even further, and more 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 and more. It turns out it actually takes 10 DNA nucleotides to make one full circle, which you don't need to know. But if I keep spinning this out for a few more rounds here and then turn this on its side, there's your twisted ladder shape. And that twisted ladder shape is called the DNA double helix. And this may be a little difficult to see, but let me show you here. Here's one DNA backbone. Here's the other DNA backbone. And there's your nitrogenous bases pairing up across the middle there. Ta-da, your traditional DNA molecule representation. So that's the double helix. That's the shape DNA molecule makes in its double-stranded form. One other point to make here, and then I believe we're done. When I've got two DNA nucleotides that are next to each other, that are paired up across the middle here, this, oops, let me get a color that I haven't used yet. Let's go brown. This right here, a pair of nucleotides that are across the middle. A pair of nucleotides across the middle here, that is called a base pair. Okay, so a pair of nucleotides that are paired up across the middle, one on each strand, is called a base pair. And we measure the length of DNA molecules in base pairs, kind of like you would measure something in centimeters or something like that. We use base pair as a unit of measurement. So so if I were to measure how long my little example nucleotide or my example DNA molecule is here, we've got one base pair, two base pair, three base pair, four base pair. So I would say that this strand here, this molecule, is 4 base pairs. We abbreviate it BP long. Notice, so a base pair has 2 DNA nucleotides in it. So this strand has 8 nucleotides, but 4 base pairs. Okay, so don't get those mixed up. 8 nucleotides, 4 base pairs. And yeah, I think that's it. If you've got any questions, feel free to let me know. You can text me or email me or your professor. Either way, feel free to set up a tutoring appointment to get help. I'm happy to help in any way that I can. thank you for watching. Sorry it was so long, and you have a wonderful day.